Next topic, lactose operon or lac operon. So lac operon is the operon that governs the metabolic degradation of lactose. It is an inducible operon. The lac operon functioning in E. coli has been thoroughly investigated by Jacob and Monod. It is a small portion of circular DNA of the bacterium. It consists of 3 structural genes and which are coordinately simultaneously induced in the presence of lactose, an operator gene, a promoter gene and a regulator gene. So 3 structural genes are there, along with that operator, promoter and regulator genes. The structural genes are Z, Y and A. They code for the enzymes beta-galactosidase, lactose permease and beta-galactoside transacetylase. Beta-galactosidase, lactose permease and beta-galactoside transacetylase. The genes transcribe together from a single promoter and form a polycystonic mRNA which directs the synthesis of the enzyme. Since these enzymes are governed by the same regulator system, they are called coordinately regulated enzymes. They are inducible enzymes produced only when lactose is present in the external nutrient medium. So lactose serves as an inducer. It inactivates the repressor system and thereby induces production of entire enzyme system necessary for lactose metabolism. Galactosidase has dual function. It mediates the breakdown of lactose to glucose and galactose and catalyzes the isomerization of lactose to allolactose, which is important in the expression of lac operon. Lactose permease remains bound with the plasma membrane and serves as a carrier molecule for the transport of lactose into the bacterium from outside. Transacetylase is believed to be involved in the acetylation of glucosides, transfer of acetyl group from acetyl-CoA to galactosides. E. coli reactions catalyzed by beta-galactosidase. When we look at that, lactose permease is there, lactose, beta-galactosidase and H2O are formed. Then galactose, glucose converted into glucose. Then metabolized in glycolytic pathway. But plasma membrane is also there, growth medium is there. Lactose later transforms to lactose permease with the help of this lactose will be later transformed to beta-galactosidase. Isomerization is formed. That will definitely lead to allolactose production, induces lac operon. That is the reaction is catalyzed by beta-galactosidase. We earlier told Z, Y, A are the 3 genes, structural genes. Then they code for enzymes beta-galactosidase, lactose permease and beta-galactoside transacetylase. Operator gene is seen close to the first structural gene, namely Z. Operator gene is seen close to the first structural gene, namely Z. It interacts with the repressor protein and serves as an on and off switch for the transcription of the structural gene to produce the enzyme. It induces or inhibits the transcription of structural genes depending upon its complexing with the repressor protein. It is active when it is free from the repressor protein and inactive when it complexes with the repressor. The inactive operator gene overlaps the promoter gene and thereby blocks the RNA polymerase. So consequently, structural genes fail to transcribe and thus enzyme production stops. Transcription by the structural gene is initiated by the promoter gene P located close to the operator gene. Promoter facilitates the binding of RNA polymerase and specifies where the transcription should actually begin. The promoter polymerase binding initiates the transcription of structural genes. A regulator gene R is located a little away from the operator gene. It regulates the functioning of the operator gene through the synthesis of a regulator protein called lac repressor. This regulation can be positive or negative. Positive regulation occurs when lactose is present in the medium and enzymes are required to metabolize it. Lactose serves as an inducer and it inactivates the lac repressor. This keeps the operator and the structural genes in a functional state. Structural genes undergo transcription and translation to produce the enzymes. Negative regulation occurs when the lactose is absent in the medium and enzymes are not needed. Now the lac operon repressor is active. It binds with the operator gene and represses it. This in turn represses the structural gene so that enzyme production does not occur. If lactose is now added to the medium, the whole system gets reversed and the enzyme production starts. Then if E. coli is grown in a medium that contains glucose but no lactose, lac operon will not be induced and only small amounts of the three enzymes will be produced. However, if the medium contains lactose but no glucose, the three structural genes of lac operon are induced and they undergo coordinate transcription and translation. As a result, the concentration of the three enzymes rises simultaneously about 1,000-fold. This is called coordinate induction. When lactose becomes completely exhausted in the medium, the transcription of the gene stops and the mRNA already produced gets degraded. So, no more enzymes will be produced and already existing ones get degraded. Allolactose, an isomer of lactose, is an intermediate product of lactose metabolism. It acts as an inducer of lac operon. It binds with the repressor, causes conformational changes in the repressor. As a result, the repressor becomes unable to bind with the operator. Consequently, the movement of RNA polymerase will not be blocked and the structural genes undergo coordinate transcription. Then catabolite repression and lac operon. Catabolite repression, also called glucose effect, is a regulatory mechanism which regulates sugar metabolizing operons in favor of glucose utilization when glucose is present in the cell. It also ensures that lac operon would be expressed at high levels only when lactose is the only source of carbon. If glucose and lactose are present together, the cell will selectively or preferentially use up glucose first. Probably the reason might be that glucose is the most ideal source of carbon and it can directly catabolized in glycolytic pathway. During this, catabolic repression inhibits lac operon with the help of a regulator protein called catabolite activator protein. This protein binds to a DNA sequence called cap binding site located upstream of lac promoter. This binding in turn favors RNA polymerase promoter binding and thereby enhances the transcription of structural gene. Cap DNA binding occurs in the presence of cyclic AMP, CAMP, whose cellular level is controlled by glucose. The formation of CAMP from AMP is catalyzed by the enzyme adenylyl cyclase, whose action is inhibited by glucose. So, availability of sufficient amounts of glucose inhibits adenylyl cyclase and thereby lowers cellular level of CAMP. As a result, CAMP DNA binding fails to occur and the structural genes of the lac operon transcribe at a very low rate until the whole genome gets metabolized. Then low availability of glucose will not inhibit adenylyl cyclase, so CAMP level will go higher. CAP DNA binding will increase and the rate of operon transcription will become maximal. When glucose and lactose are present together, catabolite repression will come into effect and the transcription of structural genes will become very low. However, once the glucose is completely used up, catabolite repression will come to an end and the transcription of structural genes will go high and all the available lactose will be used up. That is catabolite repression of lac operon. Low levels of glucose stimulates adenylyl cyclase that will bind to ATP, CAP binding site in this operon, CAP binding enhances promoter RNA polymerase binding. Then PO regions, YZA regions are there. CAP DNA binding enhances the transcription of structural genes when lac mRNA is created.